welcome from sunny Nottingham. From what I can see, I seem to be the only person out in the garden. It's my pleasure to welcome Professor Judith Breuer, who is based at Great Ormond Street in London. Judy has been leading the way with next generation viral sequencing and bioinformatics and dragging some of us more classical virologists kicking and screaming into the 21st century. And it's a real pleasure to have her come and talk to us about the efforts going on to do deep sequencing of this new virus, what that can tell us, and how we can use the data that is being generated. So I'll hand over to Judy if you take over and take the screen.
Okay, thank you very much, Will. I just need to get myself... Okay, can everybody see this? I hope everybody can see this. So thank you very much for inviting me to do this. It's a great pleasure. And I'm just going to tell you a little bit about the sequencing that's going on in the world and in the UK and its impact on public health. So just to remind you that this is the picture here on the left of the coronavirus, so-called because of the spike protein that protrudes and is very nicely shown up by the EM here. It's a positive single-stranded RNA virus. It's enveloped, and it was first described in the UK by June Almeida and David Tyrrell, so it's a UK virus. The coronaviridae can be divided up by their phylogenetic relationships into alpha, beta, gamma, and delta groups. And you can see those here on the left. The SARS-CoV-2, SARS coronavirus 2, is part of the beta, the beta family of SARS coronavirus, coronaviruses, and it's very closely related to SARS-CoV, which, as you know, circulated in the early 20s. There are other human coronaviruses, or other coronaviruses that infect humans, notably MERS, which is also a beta coronavirus. And then there are the sort of seasonal coronaviruses, and some of those are beta, and a couple of those are alpha. And what I want to show you on this slide particularly, and you may be able to see it from the left-hand side, but you'll see it better on the right, is that these viruses are very closely interspersed with animal viruses, particularly bat viruses. So coronaviruses infect many different animal species, but for the purposes of this talk, it's very important to note that the bat coronaviruses are nearby. So just to orientate you, I know you've had a talk on the virology, but this is the genome of the coronaviridae. The genome is between 26 and 32,000 bases long, and in the case of SARS-CoV-2, it's about 28,000 bases in length. And it has two main parts to it. There is the left-hand bit, the 5' end, which is the polymerase. This is the RNA polymerase that is unique to the virus. And then if you look at the SARS-CoV genome, if you see here, then the sort of structural bit of the virus has important genes that are going to be targets for vaccines. And I will talk a little bit about those in a moment. There is the spike protein here. Then there is the envelope protein, the membrane protein, and the nuclear capsid protein. And then there are a whole bunch of accessory proteins. And just to remind you that SARS-CoV-2 is very similar, 79% similar in its genome structure and its homology to SARS-CoV, the original SARS virus. So on the left, we have a picture of the pangolin, which we're now all very familiar with. I don't think I'd ever seen a pangolin before SARS-CoV-2 appeared on the scene. And just to remind you of how this all arose, actually the first case that we heard about was in December the 8th. We didn't hear about it then, but that's when the first case of a mysterious pneumonia associated with the Wuhan fish market was actually noted. In fact, the first patient was traced back to December the 1st, but not associated with the fish market. By January the 1st, the market was closed, and it was only January the 8th that the first official announcement by the Chinese state was made. So there was a month where there were rumors, but nothing official had been announced. By January the 11th, the genome had been sequenced, so that is extremely fast. And by January the 3rd, there was speculation as to what genome was similar to. And then by January the 27th, there was evidence that that genome was present in the fish market. And then by January the 31st, CEPI had already funded some vaccine studies. So just to remind you that the coronaviruses actually are not very highly mutable. So they are different to other RNA viruses. They have an endonuclease proofreading element to their polymerase, and that means that their mutation rate is not as fast as other RNA viruses. And in fact, when you look at SARS-CoV-2, it's similar to the other SARS viruses, and it has about 2.5 mutations per genome per month, and about 1 mutation between 1 and 10 transmissions. So that's much less variable than many of the RNA viruses that we're dealing with. And that has its own challenges, because of course, when we're looking at genomics, we're trying to work out whether something is more similar or less similar, and how many transmissions there have been. And if it's not evolving very fast, that can be quite challenging, and that I'll refer back to in my talk. So one of the first things that people did was to try and work out where this virus came from. And this rather complicated slide is actually not as complicated as it seems. It's about the S protein, the spike protein, the sequence of that. This is the left-hand end, the N-terminal. This is the S1 and S2 regions in the middle, and the variable loop. And what this shows you is that the nearest relative in the phylogenetic tree, which we're able to calculate because we know the mutation rate and we know the sequences, the nearest relative is actually a horseshoe bat coronavirus, shown in green. I can't see my screen very easily because I've got people in the waiting room, but anyway. And then, so that's shown there. You can see that the nearest relative to the SARS-CoV is the bat horseshoe virus. And then the next nearest relative is this pangolin virus, which is from an imported Malaysian pangolin. And in fact, that is now thought to be very closely related to another bat virus. The variable loop has a slightly different configuration. SARS-CoV is most related to the pangolin virus there, and is related to the horseshoe bat virus a little bit further away. And what that tells us is that the virus is a recombinant, and essentially what it shows us that is that although the virus is thought to be derived from the horseshoe bat coronavirus, it is probably a recombinant, or definitely a recombinant, with an unknown bat virus. Because we have the mutation rate, we can actually work out that it probably diverged from the horseshoe bat virus about 40 to 70 years ago, and it has probably been circulating in another host for all that time before crossing into humans in November or December. And the mutation rate allows us to date when all these events happened. So what has happened since it first entered the human population? Well, this is a phylogenetic tree where you can see that the virus has mutated into very many different viruses. It has a low mutation rate, but it has literally infected hundreds of thousands and millions of people now. And in that time, it has acquired mutations. And you can see it has now acquired distinct genotypes, and we're able to say now, we're able to call things by their genotypes. So from having come from 1 or 2 viruses at the very beginning, it's now diverged into hundreds of thousands and distinct genotypes. And those genotypes have arisen through the geographical spread of viruses introduced from China into other countries, and they've acquired their own mutations and become distinct genotypes. And that's very useful because we can now follow what happens to those genotypes around the world, and we can actually determine where viruses are coming from. And this is just a picture of what happened in Iceland during the month of March from a published study. And you can see these genotypes gradually entered Iceland over time and spread. And you can see that by the end of March, there were several different genotypes, each of which had been had entered the country independently. So what can we tell from this data? Well, you can tell in the UK where our first sample came from. This is a phylogenetic tree of all the viruses at a snapshot in time at the end of April. And if you map onto that, the first recorded case, the sequence of the first recorded case, you can see that it comes from a purple Japanese branch. And indeed, this was the two patients who were first positive from Chinese tourists who had to self-isolate in their hotel. So this was a Chinese virus. However, if you look at this phylogeny of European strains only, then you can see that the first virus that actually spread was this one. And you can see this is a UK virus that then spread to others. And that actually derived from an Italian virus. So those first Chinese viruses were contained, but the first virus that actually spread appears to have been an imported Italian strain. So sequencing can also be used in a more granular way to identify more local transmissions. And again, this is from the Icelandic paper, and you can see here there are clusters of similar genotypes, and this has been overlaid with epidemiological data to show trains of transmission, and sometimes where the epi seems to suggest a transmission, actually to refute that transmission. Here you have a sort of bluey, purpley dots and one orange dot. And you can look at this in more detail. This is a particular cluster. I think this was possibly in a care home or something. And in this case, the virus had actually acquired its own particular mutation, which made it very easy to track and say that these were all related. And it's estimated that 30% of transmissions are occurring in households, and that between 6 and 20% of transmissions are occurring in hospitals and care homes. So here we have the COVID-19 Genomics UK project, and this was a project that was set up in early March following the initial importing of virus into this country, and this is a sequencing consortium in the UK with many hubs throughout the UK and many more sites that are sending samples to the Wellcome Trust. So it's both local sequencing in hospitals and universities and sequencing also at the Sanger. And I should say that this figure is out of date. This was a week and a half ago. It's now over 17,000 viruses sequenced. So the UK has now contributed more COVID-19 sequences than any other country in the world. And probably COVID-19 is the virus with the most genome sequences, possibly vying with HIV, flu, and hepatitis C. Oh, I seem not to be able to. Okay. So what will COG UK tell us? Well, this is really just an overview of what we're aiming at. We hope to use the COG UK data to enhance our understanding of the epidemiology and transmission of SARS-CoV-2. So that means we can actually look at the relative contribution of local transmission versus imported cases. I showed you some of that. I'll show you some more. We can distinguish individual trains of transmission. I showed you that. We can make estimates of epidemic growth rates, unreported cases, and rates of sampling. We can determine the patterns of within-country virus spread amongst different locations and population subgroups. That will give us very good information about who is spreading it to whom, what's going to happen after lockdown, and provides insights into the mechanisms and drivers of long-distance dispersal. If you get viruses that have been circulating only in London, turning up in Nottingham, you might suppose that I've been to visit Will, perhaps, and carried my London virus with me to Nottingham. So it's going to be that sort of granularity that we can actually achieve. But we can also monitor the interventions and treatment. For example, the effect of non-pharmacological interventions, and that's lockdown and other things like that. We can look at how that affects the genotypes, and I'll show you a little bit of that. Changes in the virus antigenicity in response to the immune responses that we're all generating, and the emergence of resistance mutations in response to vaccines and drugs. And we can identify genetic markers associated with clinical severity. With the numbers of samples that we have, the power to do this is absolutely huge. And of course, this will help biological understanding and further research. So these are just a few slides to give you a snapshot of what's going on. So we can see here that immediately this is a worldwide phylogenetic tree in 3 parts. It was so big I had to cut it up into 3 parts. There are 2 major strains, both of which originated in China, A and B, and you'll see those genotypes. And then they've each diversified into others. And this shows you the blobs on this, the green, blue and red blobs are the 4 nations of the United Kingdom. And what you can see is there have been multiple introductions of these different genotypes from all over the world. So we have got local spread, but we've got multiple introductions going on. And I just need to see what's written there. So we have well over, we have about over 35 or 37 genotypes circulating. And this also gives us the data on the heterogeneity of genotypes that are circulating. And this is just the spread of the genotypes in the UK alone. Again, you can see the sort of interspersal of genotypes reflecting the fact that we all travel around a lot. But you can also see some areas in which there is actually just one colour, like here. This is a genotype that's spreading only in England. And then there are other areas, for example, at the bottom, where you can see that there are genotypes that are spreading just in Wales. And you get an even better picture of that when you look at this map. Here is your phylogenetic tree. Here is a timeline with the number of cases that have been sequenced and the number of cases tested here. More and more cases that have been sequenced. And you start to get a picture of which genotypes are spreading. And you start to get some really interesting local patterns. So here is Wales, which seems to have predominance of this pink genotype. That pink genotype is found also in other parts of the country, but it's predominant in Wales. And there are some places where there is only a particular genotype that's not been seen anywhere else. And you can imagine this gives us amazingly useful data to actually look at how things are spreading, particularly once we all start to move around. So we can also use the information in this more granular way, which is to look at local transmission. And this is a nice study that has been in preprint by Cambridge University. This is the phylogenetic tree of all the sequences that they have generated in their hospital. And here you see clusters of cases occurring in the same parts of the hospital. One of them is the dialysis unit, another one is a ward. And then they've actually gone out and got some extra data and said, well, where were these samples? Where had these people been? And some of them were inpatients who seem to have acquired it through a hospital infection. And some of them were people who were in the community and seemed to be clustered in the community. The caveat on all of this is that the genotypes are very similar, and the prevalence of different genotypes is very similar both in the community and in the hospital. And you can see that these are the genotypes along the bottom, and the numbers in the community and in the hospital are very similar. So it's quite hard to work out where people have got them from. They're living in the same community, they're admitted to their local hospital. Have they really acquired the virus in hospital, or have they actually acquired it in the community? And of course, this is all retrospective. So our plan at this moment in time, and we're just about to launch a new clinical trial called the COG Hospital Onset COVID Infection Study, which is a phase 3 prospective interventional cohort superiority study. And essentially what we are going to be evaluating is whether sequencing the virus rapidly is better able to define hospital onset COVID infections, whether we can actually work out which ones are hospital onset infections, and to measure whether producing this sequence can be used to reduce the numbers of outbreaks, because we are going to implement infection control procedures based on these sequences. We have 5 participating sites, and key to this has been the development of a reporting tool that will tell us with much more certainty whether 2 genotypes are actually linked. I'm not going to go into the details, but it's a statistical model. The readout will be high probability of linkage, medium probability of linkage, or low probability of linkage. And we will then use data on hospital contacts for each patient to work out whether they actually could have really have got this infection in the hospital. So a much more granular and possibly simpler way of looking at linked infections than using phylogeny. And finally, I'd like to just mention the fact that sequencing will have the ability to give us data on virulence, vaccine failure, and potentially disease severity. Just to remind you that the SARS-CoV-2 genome is very conserved. It's under purifying selection. And at the moment, there is no evidence that any one lineage is associated with clinical outcome or viral load differences. No resistance mutations yet described in the viruses that are circulating. And we know that there are particular vaccine targets, one of which is the spike protein. There is already a mutation that has been noted in the spike protein, and there are data suggesting that the G variant at this position is spreading faster, but this could all be due to the fact that it has just been in the country for longer, or or spread earlier in the epidemic. However, there is beginning to be some evidence of differences, potentially that the G may be associated with higher viral load. That's very confounded. It doesn't, at the moment, I don't think I believe that, but it is in a region that is very associated with immunogenicity and folding, and there are already some biological data to suggest that the 2 variants differ in their biological function. Then there are other mutations that are associated with, that have arisen more than once, the same mutation, more than once in the phylogenetic tree, so-called homoplasies. People think that that can't happen by chance, but however, when you look at that very carefully, as we have been doing, there is no evidence at the moment that these mutations that are arising in the mutations causing amino acid changes that are arising in more than one place in the tree are actually causing any difference or are due to anything other than artifact or chance. So nothing to report yet, but lots of analysis going on. And I'd just like to finish by showing my acknowledgement slide. This has been a massive amount of work by a lot of people. Thank you very much, and I'm happy to take any questions.
Thank you very much, Judy. Fantastic. I've got a number of questions here, and more will trickle through, I'm sure, but I'll try and group them together. Can we just get out of the way right at the start any concerns that this might have been a purposefully genetically engineered virus or a virus that escaped from a research laboratory? So
there is no evidence at all for that. I think the lab that was working on those bat virus, one of the viruses that is most closely related to COVID, had never been in that institute. The most likely explanation is what's happened in every other case of a new virus that has spread, is that this has emerged from a bat via another animal and into humans because of our close connection with the animal world and increasing connection.
Okay, so
but there's perfectly plausible explanation, so you don't need to invent conspiracy theories.
Okay, that will be disappointing for a certain president. Right, you talked about a number of genotypes. So for this particular virus, what kind of figure of nucleic acid dissimilarity constitutes a new genotype?
Well, I mean, this is a variable feast. It's an ongoing problem. So, I mean, you can get a new. I mean, it's very small numbers will constitute a new subgenotype. Between genotypes, there are tens to hundreds of differences between the major ones. But the new genotypes are being redefined all the time, and it's stable mutations that then are associated with onward transmission. So some new subgenotypes that are used to actually determine relationships are actually only a few nucleic acids different.
Okay, so if you track back in time through all the different variants that we see now, can you be more precise about when the virus will have crossed into humans? I mean, given that the first symptomatic case was early December, could it have been a long time before that or weeks before that?
No, there are many things that tell us it's not. First of all, you can use the dating algorithms, the modeling that tells you that the origin must have been around November, December. There's some confidence limits on that, but it must have been around November, December. And then you can look at the early trees, the early phylogenetic trees, and those are completely flat. There's no sort of branching out. They're all the viruses are sort of very closely related, and the only explanation for that is that they all evolved very recently. They're like 1 or 2 nucleotides different, so it couldn't have been any other explanation.
Okay, so if we look at the COG data, you explained how you can use the sequence to distinguish between local transmissions and imported viruses that are coming into the population. Is there any way of quantifying that and saying roughly how many of our cases have arisen de novo from entry into the country as opposed to evolution within the country?
That's ongoing. That exact analysis is now being done, and there's a real effort to try and actually quantify that, because obviously it's going to be very important going forward to understand, if things take off, where it's all coming from. I don't have that answer now, but I know that analysis is underway.
So, as we discussed earlier on, Australia announced their 100th death today, which is staggering compared to ours, which does beg the question whether they were very quick to stop travellers coming in. And this, you may not wish to answer this, but do you think it would have been a good idea to have done something about that sooner than we still haven't quarantined travellers?
Well, I think the message is that we need to do, so acting fast is good, and we need to use all the modern tools that we have in order to get on top of this virus. And I mean, Australia didn't have to use sequencing because it acted very fast, but they also used testing and tracing and quarantining very fast. And testing and tracing is just a modern tool that we have available now. I mean, if you go back to the 1918 flu pandemic, they were using, blunt instrument quarantining to stop 1918 spreading, and we now have the option to actually do it in a much more focused way by actually working out who should quarantine, not having to quarantine everybody. But the problem is, of course, when you've got too many infections spreading, you haven't got the resource. I mean, it's just overwhelming. You can't actually do that. So yes, if we'd managed to do this more modern and 21st century approach to quarantining at the very beginning, then I think we would have been in a much better position. But yeah,
okay.
But that's all with hindsight. We can all say that.
Have you ever thought of becoming a politician? So somebody has asked about the low mutability of this virus, and I have to say I've been surprised for an RNA virus at how infrequent sequence change happens. So the basic question is this a good thing or a bad thing as far as the infection in humans is concerned?
Well, in some ways, it's a good thing. I mean, what it does tell us that an awful lot of the variation that we're seeing in clinical outcomes is about the host. So we really need to actually get to grips with what's going on in people. It's been a massive experiment. I mean, it's a more or less the same virus released into the human population with different phenotypes evolving. And those phenotypes are largely due to human genetic variation. And I should mention that there is another part to the COG study, which is sequencing the genomes, the human genomes of people whose viruses have been sequenced. So, yeah, I mean, I think that in some ways it's a good thing, but it's, you know, I can't see that it's a particularly bad thing to have a virus that is very stable. Whether that means we can make better vaccines, I don't know. I mean, I'm not sure that it necessarily does. I mean, it's, you know, and also there is quite a lot of variation now. So we're beginning to see, you know, variants in key proteins. And yeah, so who knows whether it's going to be good or bad in the end. We just don't know.
Okay, you did refer to drug resistance mutations, and clearly we don't have any drugs that we know work. Is the fact that you're not seeing a significant mutation in presumably the polymerase good evidence that the drugs that we're using simply don't have any effect?
Well, I think we're not actually sampling properly to look for drug resistance. I mean, we're not seeing circulating mutations that we would actually attribute to drugs. But of course, drugs have not been very widely used. And we're only doing one sample from each person. And I think if you're going to try and look for drug resistance, you need to take the sample at the end of treatment and have a look and see whether that has acquired mutations. And there's been very little of that done, very little longitudinal sampling and sequencing to look at the acquisition of mutations. Now, of course, those viruses that are at the end of treatment are probably not then spreading. So there'd have to be an awful lot of drug resistance before you started to see it spreading in the population. And we're just not using very many drugs, as you say, because we just don't know which, if any, will work.
Yeah, so actually the persistence of PCR positivity is also an issue that's coming up. So we've seen patients who remain PCR positive for weeks. So are you studying inpatient evolution in that way?
I'm studying it. It's not something that COG UK is doing, but I've had a long interest because when my clinical practice is at Great Ormond Street, we have very sick children who die of persistent, common viral infections, common respiratory viral infections, and develop, you know, all sorts of resistance. And we treat them with the same drugs that we're now using for COVID. We treat the children. So I've had a long-standing interest, and I'm now deep sequencing COVID as well to see whether over time there is the acquisition of mutations that could be associated with resistance.
Out of interest, have you had many patients at Great Ormond Street?
We've had about, I think, around 30, very few, and that's the whole of, you know, northwest London and the surrounding area. So very few children affected, but this very interesting COVID inflammatory syndrome emerging in children, which looks very much like Kawasaki.
Yeah, very interesting. I'm going to read this question out verbatim because it's not information that I'm party to. So someone has typed, there are studies that confirm the virus has mutated 33 times and the first cases were in October.
I can't hear you. I can't hear you. Didn't hear that. It confirmed it's mutated.
That the first cases were in October rather than December. Is this information correct? And the person says, I think I read the first case was from Italy.
No.
I don't know if that means, no.
No, I mean, the first case in the UK probably came from a person who came back from Italy. It's the first case that spread in the UK probably came from a person who'd been in Italy rather than someone from China, since which time we have had Chinese cases coming in and spreading in this country. The dating, as I'm aware of it, is back to November, December. If you look at the early phylogenetic trees and you calculate, they have, you know, the early viruses are all 1 or 2 mutations apart, and you calculate the time that it must have taken. It can't have been more than a month previously that they came into humans.
Okay. Do you have any data on evolution in tissue culture? Are there any adaptive mutations that improve replication?
There has been at least one mutation, one deletion described in tissue culture, which seems to be an adaptation to tissue culture. And that has also been found in 1 or 2, very few, circulating viruses. I can't remember which gene it's deleted in, but there are, and indeed, both SARS-CoV and SARS-CoV-2 do acquire deletions in different accessory genes generally, and they seem to be stable deletions.
Okay, I think we're coming towards the end here, but as a general question, in your experience of virus evolution, this virus is clearly here to stay. Is it likely that it will evolve to be less pathogenic and become just a common cold virus? Or is that wishful thinking?
Who knows? I mean, you know, how long does it take? That's the other thing. I mean, our history is that we think that that's what happens to viruses, but that happens over millennia, you know. Whether this will happen in our lifetimes, the only thing I can think is that if infection occurs mostly in childhood, if we get to the stage where everybody is immune and the birth cohort is the only one that is not immune, then infections will occur in childhood, and that's presumably what happens with most coronaviruses, and children are not as badly affected. And so it may become something that is, you know, a mild, one of those children's viruses that causes, you know, mild infection, and everybody else has already had it and is not, you know, is not susceptible to the most, you know, to it being very severe. So that's one potential outcome. Whether it evolves very fast to become less virulent, I don't know. I mean, I think it's unlikely in our over our lifetimes.
Okay, so I will make this the very last question. It is in fact the last one I've received. You've sort of touched on this, but the question is, will the level of mutation affect the efficacy of vaccines?
But potentially, I mean, who knows whether this DG614 mutation may affect the vaccine. I mean, there is the potential for that to happen, and which is why it's very important to really have a handle on what the variation is before we start vaccinating, and then to monitor, to use the sequencing to monitor the evolution of vaccine escape mutants if those do arrive. So it is potentially possible.
Okay, well, it would be nice to think we had a vaccine that we could actually experiment with.
Absolutely.
Hopefully soon. So on that note, I'll conclude this evening's seminar. Thank you ever so much, Judy, for taking the time to educate us in such a clear way and to withstand my interrogation. And with that, I'll just say goodbye and see you again next Wednesday. Thank you very much.
Thanks very much.